Okay, yeah. Well, thanks. It's a very interesting meeting and incredible scope and breadth of things to think about. And so I'm going to give you actually some things to think about of really when I was invited to speak about this, to ask about how research advances were furthering knowledge and how that was impacting what we variously refer to as precision or personalized medicine, in particular, cancer. And so I'm going to try to talk about two things. And the first one is this notion that research advances really drive new knowledge of mechanisms of cancer, and this can lead to innovative cancer therapies. And also the second point about better tools and this new knowledge are really then driving this frontier of personalized cancer medicine. So what is cancer? Well, it's a disease of extraordinary complexity at all levels. It is a genetic disease, and every cancer has mutations in the genome. And again, we've already heard about how genome sequencing is really informing us in great depth about the alterations in the cancer cell genome. But it's also diverse histologically, pathologically, prognostically, response to therapy. So it's rather overwhelming across the spectrum of human cancers, all of this complexity. And so the issue comes, can we rationalize this complexity based on really an avalanche of information about cancer at all these levels? And the question is, are there common principles underlying this diversity? And so basically these research advances, which of course we don't have time to discuss, have revealed underlying principles. And that is that disparate cancers share fundamental qualities, and that these distinctive capabilities are acquired aberrantly by cancers as they develop and progress to pathologic and lethal malignancies. And arguably, these acquired capabilities collectively explain the manifestation of the disease. So this is at sort of the 30,000-foot level. So we've dubbed these the hallmarks of cancer, these acquired capabilities that we think underlie the progression of the disease and also the diversity of it. And so I'm just going to walk you through those as sort of a conceptual overview of cancer, particularly for those of you who are not specialists in this field. And the first hallmark is of sustaining proliferative signaling, because cancer is a disease of aberrant and continuing and inappropriate cell proliferation. And so the notion is that the accelerator that drives the cell division cycle is on. It's full speed ahead. The cells are instructed to grow and divide chronically, whereas normally in the body, cell growth is carefully orchestrated. The second hallmark involves evading growth suppressors, that not only are there signals that drive proliferation, there's also brakes that turn off proliferation, because that's also fundamental to the fact that we grow from a single cell to one of hundreds of millions of cells, and then we stop growing in most cases. And so there are cell intrinsic and extrinsic brakes that limit proliferation in normal cells, and these have been failed or abrogated in most cancers, and these stop signals become disabled. A third capability involves the resisting cell death, because it turns out that all of our cells have inborn mechanisms for assisted suicide, of which there's a number of them. And basically it's meant in part to cull out aberrant cells. But what we see in symptomatic cancers are ones that have learned how to avoid this assisted suicide, and they abrogate this inborn willingness of cells to die for the benefit of the organism. The fourth hallmark involves a replicative immortality, of the ability to grow forever, because embedded and integral to the structure of our chromosomes is a counting mechanism that counts the number of cell divisions by the length of short repeats at the end of the chromosomes. And this is a counting mechanism that after a normal cell is divided a certain number of times, it basically triggers stop signals, which cause the cell to either stop proliferating or to die. And basically, cancer cells learn how to evade this counting mechanism and convey upon themselves this replicative immortality. And they do so largely by activating a specialized enzyme called telomerase, which actually counteracts this, this counting mechanism. It was a Nobel Prize winning discovery some years ago. The fifth capability or hallmark involves inducing new blood vessel growth, the process of angiogenesis, which turns on new blood vessel growth to feed and nurture this growing mass of cancer cells. They need oxygen, glucose, and other blood-borne metabolites. The sixth capability or hallmark involves activating the capability to invade locally and metastasize to different organs, which really underlies much of cancer lethality. And the cancer cells become guerrillas that grow by migrating and invading into normal organs locally and throughout the body. And they do so because they can get access to a new fertile soil in which to grow. And so they're really an invasive weed, if you like to think about our discussion yesterday. And a new notion is really that, and we've already heard a bit about metabolism, but cancer cells deregulate cellular energetics and metabolism to really fuel continuing proliferation rather than the normal homeostatic functions that most cells have. And so they activate alternative energy sources. They sort of become hybrid engines where they can use multiple sources of fuel and pathways to drive cell growth and also to produce all the cellular building blocks for new cancer cells. And the eighth hallmark involves avoiding immune destruction, that we now appreciate that the immune system, in many cases, recognizes that cancers or tumors are aberrant and seeks at some stage of disease progression to try to attack it. But what we see in symptomatic cancers is that they develop a variety of mechanisms that allow them to avoid this attack and killing by immune cells. And this has actually fueled a whole revolution now in activating the immune system to kill tumors. And so this is a very exciting time, and this is therefore, we think, a crucial mechanism that many cancers engage. So these ideas came out of just this wealth of new knowledge about cancer mechanisms. And so the hypothesis based on these research advances is that these 8 hallmark capabilities are sufficient to generate a lethal cancer, and that you can see across the diversity of human cancers differing ways and means by which these capabilities are activated. And another key concept, although cancer is a genetic disease, and so the field is just full of studies on the cancer cells which contain the aberrant mutations, and we're seeing, and you've already heard about resequencing of cancer cell genomes to identify all the aberrations. And there's actually a school of thought that says that we can be fully informed about cancer simply by understanding or interrogating the cancer cell genome and identifying the mutations. But I believe that's going to be simplistic, because there are other epigenetic factors that are involved above and beyond mutations of the cancer cell genome. And one of those is that the tumors really are not bags of cancer cells, even though that you could be led to that assumption. That they're actually outlaw organs that have a heterotypic collection of interacting cells that contribute in really fundamental ways to the success, the aberrant success of cancer. And so in addition to various flavors of cancer cells, which again have genetic mutations in them, we have a variety of other cells which are functionally important but are not themselves mutated. And these include endothelial cells and periocytes of the tumor vasculature, cancer-associated fibroblastic cells, and inflammatory cells of the immune system that are actually promoting, not attacking the tumors. And these are principally cells that are involved in wound healing, which is another function of the immune system, quite different from the attack that we see from cytotoxic T cells. And so these wound healing cells actually stimulate many of the hallmark capabilities and really are accessories to the crime here. And so all of these cells are functionally contributing to what we see in symptomatic cancer and need to be factored into really the knowledge of cancer and potentially the translation to better therapies. So basically, yeah, so the notion then is that these research advances have produced new knowledge, which has led to, conceptualizations such as this one on these acquired capabilities or the hallmarks of cancer. And arguably, this is really helping, to rationalize and conceptualize all of the research advances that are forthcoming from the research community. But the ancillary question is whether this concept is applicable to the goal of more effectively treating human cancer. And so I'm just going to leave you with a few thoughts there. First of all, it turns out if you take all of the 8 hallmark capabilities plus a couple other factors which help, which facilitate their acquisition, which include, for example, genome instability and mutation, that there are targeted therapies, so-called smart bombs, that really target each of these capabilities. And so that's really exciting. It says we have a whole pharmacopoeia of drugs that can target each of these distinctive capabilities. And so what is it? And so is that affording new opportunities? Well, let me just give you an example of the most obvious one that the field's been focused on, and that's targeting the proliferative signaling hallmark, and with a whole series of highly selective drugs that are targeting differing mechanisms of driving this proliferative hallmark. And I'll give you an example here of an inhibitor called Braf that inhibits proliferative signaling. And it does so in a disease called melanoma. Now, metastatic melanoma has been virtually treatable. It metastasizes around the body, and this is just an image of a patient. These are the normal organs. This is the multiple metastases. And so these metastases, about 50% of melanoma, contain a mutation in a gene that drives proliferative signaling called BRAF. And there have been very selective inhibitors for BRAF that have been developed. And they do what we've dreamed about for metastatic disease, which is it melts away. So again, these are normal organs. This is all essentially this multifocal metastatic disease has melted away in a couple weeks. This is a patient that was treated with this drug. And so this is really the dream that this is what we dream about, really, for a highly effective cancer treatment. And so that's the good news. The bad news is that this is transitory. So 6 months later, multifocal metastatic disease is back with a vengeance, even more aggressive. And so these are like the weeds we were talking about yesterday. I mean, these evolve, the tumor, this is Darwinian evolution. These cancer cells are knocked back, but they find a way to adapt and escape, and they recur. And so that is the challenge, the reality check that we're facing, is that targeting single hallmarks is most often transitory, followed by adaptive resistance and relapse. So now, looking to the future, we're thinking about new ways to approach this reality, and one of which is sort of what I would call a new battle plan for future therapy, which involves targeting these distinctive capabilities, by air, by land, and by sea. If you target one, you get adaptive resistance, but what if you target multiple ones? And so, for example, you can target, you can leverage the immune system both by activating cytotoxic T cells by taking out this evading immune destruction hallmark, but also removing these tumor-promoting, wound-healing inflammatory cells, which also have the capability to contribute to this immune evasion. And so we can leverage, for example, several hallmarks to activate the immune system, or we can target proliferative signaling and really stimulate cell death in combination. These are just examples of how you could proceed. And so I think the future could well involve a sort of a battle space plan where we take complex therapeutic regimens, which are enabled not only by knowledge, but also by better tools to detect non-invasively in liquid biopsies and by better imaging technologies, the recurrence of disease and potentially the mechanisms of adaptive resistance. So you could start off with immunotherapies, go into these oncogenic drivers that I just showed you. Angiogenesis and metastasis are also targetable. So you can imagine, but again, Darwinian evolution says that in many patients, you treat them with one combination, that may not be enough, and so you can then switch to another. So this is just a sort of a conceptualization of where I think that the future may take us in terms of producing more enduring therapies. So then notably, we have better diagnostic tools as well, and that's enabling the identification of the precise hallmark enabling mechanisms and the particular adaptive resistance modalities that are operative in individual patients. And therefore, which is allowing us now the opportunity to use the best therapies from this growing pharmacopoeia to treat particular patients, because this diversity that I described, even if they've all acquired the same capabilities, they use different genetic and epigenetic mechanisms, but we now have the capability to really interrogate an individual's tumor and decide how that should be treated. And this is sort of the concept of precision or personalized medicine. And so I'm going to just give you a few thoughts about that. I'm actually adapted a presentation of my colleague in Lausanne, Olivier Michelin, who's heading a personalized medicine initiative there. And so I just borrowed a few of his slides. And so the key point is that personalized approaches are become mandatory for many cancer types because of this advancing knowledge of mechanism. We no longer treat lung cancer as lung cancer. There are multiple subtypes of non-small cell lung cancer, largely caused by smoking, and they have differing activating mutations that drive the proliferative hallmark, and depending on which mutation you have will dictate which drug you're going to be treated with. And essential for these analyses is actually having biopsies and other means of interrogating tumors, both before treatment and when they develop adaptive resistance. And that's really important because not only are there multiple primary mechanisms that drive tumor growth and progression, but there are many mechanisms of adaptive resistance. So for example, this Braf oncogene that I just described and showed you that it had brilliant activity, but then very rapid recurrence due to adaptive resistance. Well, here's just, it is in a network of other signaling molecules, signaling genes that are altered during adaptive resistance. And so these are the 20 most frequently altered genes that have been demonstrably shown to contribute to adaptive resistance and relapse in BRAF melanoma. So it's not enough just to target BRAF, but again, which of these do you target? You need to know in a particular patient's tumor which of these adaptive resistance mechanisms have been co-opted. So we're shifting from a single target, single escape mechanism to an integrated systems biology of response and escape. And as already been alluded, there are a whole series of diagnostic tools that can inform us about tumors. And so there's clinical data, environmental data, genomics from the tumor and from circulating DNA, immunohistochemistry, proteomics, and metabolomics. So we increasingly have a broad set of research tools that allow us to interrogate tumors, as I said, both prior to treatment as well as during the adaptive resistance phase, which seems to be virtually inevitable. And you can integrate this data and use that to guide clinical decisions. And so, but clearly one of the hot areas right now is detecting gene mutations, both in baseline and adaptive resistance. And so that's being, combined with clinical data, and we collect tumor cells, and increasingly now also looking at the cells in the microenvironment that I alluded to. And so you can do molecular analyses that include not only genome sequencing, but also of the expressed genes, the so-called transcriptome, and how that's changing in cancer cells and in resistant cancer cells. And then this can help guide first-line targeted therapies, and you can then monitor the responses and survival in response to those. And then when you have failure and progression, you can switch to other therapies. And those switches can be, again, guided by personalized approaches of analyzing the resistant tumor to understand what of these panoply of mechanisms it's actually utilizing so that you can target it appropriately. But one of the other really, I think, exciting things in this area right now is that it's one thing if you have a cancer, right now you go to one of the top medical centers in the world if you're smart, and you go to the Karolinska, or you go to MD Anderson or Sloan Kettering, or you go to ours in Lausanne. But what about all the patients who are out in the community hospitals and private clinics who aren't so close to a major medical center? Well, I mean, basically now there are sort of next generation tumor boards are being created with video conferencing that is really allowing precision diagnosis and personalized therapy regionally to bring in the broader population. Because the situation we faced is that many of these community doctors aren't aware of the latest advances in cancer mechanisms, the latest new therapies, and also the notion that you can't blindly treat tumors. You have to know what's going on in that tumor. And so now really, you have your local oncologist and your patient, but you can do high throughput sequencing of that tumor, and then has already been alluded to in other diseases, you need a whole team of people, oncologists, geneticists, pathologists, bioinformaticians, who are helping to interrogate this and other information that you have about the patient, and you can do this into a web-based system, which then allows this to be communicated. And so I'll just give you an example of that. So what Olivier and his colleagues are doing is establishing a network throughout Western Switzerland of around 2 million people that brings in all the community hospitals and private clinics scattered around the region to bring them up to speed with the latest therapies. Because we have two major centers, the CHUV in Lausanne and Axel in Geneva, that are really state of the art in terms of cancer care, but these others are not. But now the oncologists and their patients in these other centers are being integrated into a web-based tumor board. And a lot of this is happened remotely, but there's also data managers who go around and visit all these sites. And so the way this operates is the idea is that these patients do not want to go to Lausanne or Geneva in this case to be treated on a routine basis, but they do want to know what to be treated with. And so you basically have your regional hospital, you collect blocks and slides and blood samples, and those are then interrogated, sent to the major centers, and they're interrogated, and then and established with electronic patient records, and all of this is then integrated and analyzed. And then patients, as appropriate, can be put into clinical trials of new therapies, depending on the nature of their disease. And the patients can come in selectively to the major centers to be treated, but then they're managed in their local hospital next to where they live. So this is, I think, an exciting, and I think this is what we're going to see worldwide as this whole notion of precision medicine evolves. But it does require a broad expertise. It's no longer just an oncologist and the patient, and because we're applying all these analytic techniques now of genome sequencing, but also histopathology, liquid biopsies in the blood, and so on and so forth. And so this is really an exciting frontier, but you need a whole team of specialists. I mean, as I said, it's no longer just the oncologist, but you need geneticists, bioinformaticians, and biologists, and so on. But that's going to then allow really more informed treatments of patients with greater prospects for response. And so I'll just give you one example of how this sort of precision medicine can have clinical benefit. And so because what you can find is in genome sequencing of a patient are, for example, unusual actionable mutations. And so this is an example of a urothelial carcinoma of a patient who'd failed all the standard therapies. And genome sequencing revealed a mutation in a gene called HER2, which is actually activated in one major class of breast cancer, but is not typical in urothelial carcinoma. But the adaptive resistance mechanism in this patient involved in part activation of HER2. But there are drugs which target HER2, so this patient could then be put on trastuzumab or Herceptin, which targets this mutant protein. And so this is just an example of how precision medicine can, this patient otherwise would have no prospects for any kind of meaningful therapy. And so this is just an example of where, you know, the future is going to be with this precision medicine, that we can really guide patients toward clinical trials, even if they have a rare mutation. So, you know, I think this is really important, because what we're finding with this diversity of mechanisms, that you can have a cancer type for which there's an actionable mutation like this, but it's not typical, and it's in most patients. And so you can be sort of N of one of somebody who can have a response to these drugs. I actually have an old friend in San Francisco who has metastatic breast cancer, and was put on a trial of an angiogenesis inhibitor because she had no hope for anything. And she's one of one person out of hundreds who responded, and she's disease-free 5 years later when she had little hope. And so this is, I think, the future of precision medicine. So basically, yeah, the take-home messages are that research advances are producing new knowledge of mechanism, which are leading to innovative cancer therapies, and these powerful analytic tools, including inexpensive and rapid DNA sequencing, buoyed by new knowledge, are really enabling this exciting frontier of personalized cancer medicine. Thank you.
We have time for one question before the break. Bjorn?
Thank you for a very exciting lecture. Yeah. How do you think AI will support what you're saying? I'm not implying that it will replace the expert panels or anything, but we know the Watson and IBM initiative. What's your view on?
Oh, I agree, absolutely. And in fact, that is, you know, I was telegraphing through all this, but indeed, you know, and analyzing all this data and putting it all together, I mean, this group is actually collaborating with this IBM Watson enterprise to try to use AI and machine learning to help guide these decisions. No, so I think this is definitely part of the future.
Yes, one more.
Anna Sandström from AstraZeneca. So we are truly seeing this, and that's in our strategy as well. I mean, one of the recent collaborations that we have gone into is combining then a PARP inhibitor with immuno-oncology with another company, Merck. And that type of collaborations, I think we will see more and more, since we see that we need to attack cancer through many of these pathways. And the other thing is that we also see the resistance development, and if we have the link between the clinical data and early on catching that we have a resistance developing, we can go back to the lab and create a new drug, and actually do that much, much quicker than the whole line. And then it's the last line of treatment, and it makes it much easier to also try to bring that to patients in a very short timeline. We also have one of those examples where we within 3 years had a new drug after resistance development.
I think, because again, that is the great opportunity now with all these tools to really analyze resistance mechanisms, because it is so important. So I think, and I agree completely also with this whole, I believe that these distinctive combined therapies are going to pay off in more enduring therapies. And yeah, so it's an exciting time. Thank you.
Okay, thank you again for the wonderful presentation.